Mutualism is a symbiotic relationship between two species in which each benefits. 27. In this type of relationships, each species involved provides some services to the other species, and this could be nutrition, defense, etc. 29. Signaling is known to play a major role in selecting mutualistic partners. Odors and chemical detection is involved in the formation of symbiotic relation between most fungus-growing insect. The attraction of ambrosia beetle to its fungal symbionts indicate that some signaling by microbial volatile organic compound S, MVOCs, produced by the fungi leads to this partner choice. 5. In ambrosia symbiosis, the ambrosia beetle obtains nutrition from the ambrosia fungi while the fungi relies on its host, ambrosia beetle, for its dispersal and cultivar maintenance. 30-31. Another common mutualistic relationships include cleaning symbiosis, animal-induced pollination, or protection from predators. One example of insect mutualism is the pollination of flowering plants by insects, a field of study known as entomology. Primarily, various bee species work as pollinators of flowering plants, feeding on their nectar and in turn picking up their pollen and spreading it to other flowers. 27. Another example of insect mutualism is the relationship between ants and aphids. The aphids provide food, honeydew, for the ants, and the ants in return protect the aphid from its predators by feeding on the predators. 32. Another notable example is the relationship between aphids, Hemiptera, aphididae, and their symbiotic bacterium Buchnera aphidicola. These microbes are situated within specialized internal structures known as bacteriomes, within the insect's body. 33. Since aphids' diet of plant sap lacks certain essential amino acids and vitamins, Buchnera supplies these nutrients to them. In exchange, aphids provide Buchnera with a protected environment within specialized cells, as Buchnera's genome is highly reduced, rendering it incapable of surviving independently. 34-35. Mites benefiting from the movement of Nichrophorus humator. Commensalism. Commensalism is a different type of ecological interaction between species in which one species gains benefits while the other is neither harmed nor benefited. 7. Two examples of commensalism that can be seen in insect ecology are phoracy, an interaction in which one attaches itself to another for transportation, and inquilinism, the use of another organism for shelter. Ticks and mites have adapted to latch onto beetles, flies, and bees, as well as other organisms, for transportation, an example of phoracy. 36. In terms of inquilinism, insects commonly establish themselves in human garages or shelters of other animals for protection against predators and weather. Parasitoid insects. Parasitoids are insects that live intimately with a host, feed off of the host like a parasite, but eventually kill the host. This specific type of species interaction is exclusive to insects and is employed most commonly by wasps. An example of this is when parasitoid wasps inject their eggs into aphids. The eggs will eventually hatch and produce wasp larvae that feed on and consume the organism. Additionally, some parasitoids chemically affect the host to propagate the development of parasitic offspring. Parasitoid wasps typically prey on a specific insect or spider species, and the host life stage at which the wasp deposits its seed differs. In regard to humans, parasitoid insects are favored because they can be used as biological pest controls for farmers, preying on other insects that damage crops. 37. Competition. Insects often compete with each other for resources such as food, territory, and mates. Competition can occur within species, intraspecific, or between species, interspecific. This competition can lead to adaptations and niche differentiation, where species evolve to occupy different ecological niches to minimize competition. Neutralism. In some cases, insects may interact with each other without affecting one another positively or negatively. They simply coexist without any significant impact on each other's fitness or survival. 38. In such a relationship, the interaction that happens between the organisms involved are always indirect or incidental. 6. This type of relationship is often observed when insects occupy different habitats or have minimal interactions. 38. Another example is when a bird uses a tree as a lookout without having feeding on the plant or insect on the plant. 39. An example of such relationship is the one that exists between a tarantulas and a cacti that live in a barren region. 6. Facilitation. Facilitation occurs when one species indirectly benefits another species by modifying the environment. For example, certain insects may create microhabitats or modify resources that become beneficial for other insect species. An example of this could be a species of insect that creates shelter or nesting sites that are subsequently utilized by other insect species. 40. Amensalism. Amensalism is a non-symbiotic species interaction in which one organism negatively affects the other organism but is unaffected by that organism. This type of species interaction is common in nature, and an example in insect ecology is between goats and insects. The two individuals compete for the same food source, but goats will deprive the latter from feeding. 41. The goat is completely unaffected by the interaction, but the insect is left hungry. Mimicry. Insects may evolve to mimic the appearance, behavior, or other characteristics of other species. This can be beneficial for the mimicking species in various ways, such as gaining protection from predators or gaining access to resources. For example, some harmless insects mimic the appearance of more dangerous or unpalatable species to avoid predation. 42. Allelopathy. Allelopathy involves the release of chemicals by one species that affects the growth, development, or behavior of another species. While this type of interaction is more commonly associated with plants, certain insects may also engage in allelopathic relationships with each other. These chemicals can influence competition, reproduction, or survival of other insect species in the vicinity. 43. Coevolution. Coevolution is the ecological process by which two species exclusively affect each other's evolution. This concept is essential to the study of insect ecology. Coevolution is particularly important in how it can lead to both micro and macroevolutionary changes. Microevolutionary changes include shifts in genome and alleles, while macroevolution is the emergence of a new species, also called speciation. 44. Two species that coevolve experience reciprocal evolution and go through biological changes as a result of the other species. 45. One example of this in insect ecology is the coevolution of Dicyscolia ciliata, a species of wasp, and Ophrys speculum, a species of orchid. These two species have both evolved in such a way that the wasp is the only known pollinator of the plant. This relationship can be seen in other species of flowering plants and pollinating insects, but a more distinct example is the coevolution of ants and acacias. The acacia ant, Pseudomyrmex ferruginea, is an insect that has been discovered to protect 5 different species of acacia trees. The ant provides protection to the plant while the acacias reciprocate by supplying food and shelter. Over generations, these two species have adapted to accommodate each other, an example of coevolution. Role. Insects play numerous roles which have both negative and positive impact on the environment and other organisms. These roles include pollination, seed dispersal, improving soil fertility, act as a source of food for other organisms, and involve in disease transmission. Pollination and seed dispersal is known to be mostly caused by animals, and insects are responsible for about 85% of pollination in angiosperms. 46. Ants are known to cause dispersion of the seeds of more than 100 species of plants found in Brazilian Caatinga. 47. Soil-dwelling insects such as ant, termites and beetles help in the decomposition of plant residue, aeration of soil, and enhancement of soil structure. These activities help in improving soil health and fertility. 48. Insects again serve as a source of food for other organisms such as reptiles, bird, amphibians, fishes, and many others. 49. Insects are also involved in the transfer of vector-borne disease, which accounts for approximately 700,000 deaths annually. Insect morphology is the study and description of the physical form of insects. The terminology used to describe insects is similar to that used for other arthropods due to their shared evolutionary history. 3 physical features separate insects from other arthropods. They have a body divided into 3 regions, called tagmata, head, thorax, and abdomen, 3 pairs of legs, and mouthparts located outside of the head capsule. This position of the mouthparts divides them from their closest relatives, the non-insect hexapods, which include Protura, Dipleura, and Colembala. Insect morphology. Legend of body parts. Tagmata: A. Head, B. Thorax, C. Abdomen. 1. Antenna. 2. Ocelli, lower. 3. Ocelli, upper. 4. Compound eye. 5. Brain, cerebral ganglia. 6. Prothorax. 7. Dorsal blood vessel. 8. Tracheal tubes, trunk with spiracle. 9. Mesothorax. 10. Metathorax. 11. Forewing. 12. Hindwing. 13. Midgut, stomach. 14. Dorsal tube, heart. 15. Ovary. 16. Hindgut, intestine, rectum and anus. 17. Anus. 18. Oviduct. 19. Nerve cord, abdominal ganglia. 20. Malpighian tubes. 21. Tarsal pads. 22. Claws. 23. Tarsus. 24. Tibia. 25. Femur. 26. Trochanter. 27. Foregut crop, gizzard. 28. Thoracic ganglion. 29. Coxa. 30. Salivary gland. 31. Subesophageal ganglion. 32. Mouthparts. There is enormous variation in body structure amongst insect species. Individuals can range from 0.3 mm fairyflies to 30 cm across. Great owlet moth, 1.7 have no eyes or many, well-developed wings or none, and legs modified for running, jumping, swimming, or even digging. These modifications allow insects to occupy almost every ecological niche except the deep ocean. This article describes the basic insect body and some variations of the different body parts. In the process, it defines many of the technical terms used to describe insect bodies. Anatomy summary. Insects, like all arthropods, have no interior skeleton. Instead, they have an exoskeleton, a hard outer layer made mostly of chitin that protects and supports the body. The insect body is divided into 3 parts: the head, thorax, and abdomen. 2. The head is specialized for sensory input and food intake. The thorax, which is the anchor point for the legs and wings, if present, is specialized for locomotion, and the abdomen is for digestion, respiration, excretion, and reproduction. 122-48. Although the general function of the 3 body regions is the same across all insect species, there are major differences in basic structure, with wings, legs, antennae, and mouthparts being variable from group to group. 3. External. Exoskeleton. The insect's outer skeleton, the cuticle, consists of 2 layers: the epicuticle, which is a thin, waxy, water-resistant outer layer that lacks chitin, and the layer under it is called the procuticle. This is chitinous and much thicker than the epicuticle and has 2 layers. The outer is the exocuticle while the inner is the endocuticle. The tough and flexible endocuticle is built from numerous layers of fibrous chitin and proteins, crisscrossing each other in a sandwich pattern, while the exocuticle is rigid and sclerotized. 122-24. The exocuticle is greatly reduced in many soft-bodied insects, especially the larval stages, e.g., caterpillars. Chemically, chitin is a long-chain polymer of an acetylglucosamine, a derivative of glucose. In its unmodified form, chitin is translucent, pliable, and resilient. In arthropods, however, it is often modified, becoming embedded in a hardened proteinaceous matrix, which forms much of the exoskeleton. In its pure form, it is leathery, but when encrusted in calcium carbonate, it becomes much harder. 4. The difference between the unmodified and modified forms is evident when comparing the body wall of a caterpillar unmodified to a beetle modified. From the embryonic stages, a layer of columnar or cuboidal epithelial cells gives rise to the external cuticle and an internal basement membrane. The majority of insect material is inside of the endocuticle. The cuticle provides muscular support and acts as a protective shield as the insect develops. However, since it cannot grow, the external sclerotized part of the cuticle is periodically shed in a process called molting. As the time for molting approaches, most of the exocutical material is reabsorbed. In molting, the old cuticle separates from the epidermis, apolysis. Enzymatic molting fluid is then released between the old cuticle and epidermis, which separates the exocuticle by digesting the endocuticle and sequestering its material for the new cuticle. When the new cuticle has formed sufficiently, the epicuticle and reduced exocuticle are shed in. Ecdysis. 5:16-20. The 4 principal regions of an insect body segment are the tergum or dorsal, sternum or ventral, and the 2 pleural or laterals. Hardened plates in the exoskeleton are called sclerites, which are subdivisions of the major regions, tergites, sternites, and pleurites, for respective regions tergum, sternum, and pleuron. 6. Head. Head of Orthoptera, acrididae. A. Antenna. B. Ocelli. C. Vertex. D. Compound I. E. Occiput. F. Genna. G. Pleurostoma. H. Mandible. I. Labial palp. J. Maxillary palps. K. Maxilla. L. Labrum. M. Clypeus. N. Frons. Larva of beetle, family Cerambicidae, showing sclerotized epicranium, rest of body hardly sclerotized. Larva of syrphid fly, member of Cyclorrhapha, without epicranium, almost without sclerotization apart from its jaws. The head in most insects is enclosed in a hard, heavily sclerotized, exoskeletal head capsule. The main exception is in those species whose larvae are not fully sclerotized, mainly some Holometabola, but even most unsclerotized or weakly sclerotized larvae tend to have well-sclerotized head capsules, for example, the larvae of Coleoptera and Hymenoptera. The larvae of Cyclorrhapha, however, tend to have hardly any head capsule at all. The head capsule bears most of the sensory organs, including the antennae, ocelli, and compound eyes, along with the mouthparts. In the adult insect, the head capsule appears unsegmented, though embryological studies show it to consist of 6 segments that bear the paired head appendages, including the mouthparts, each pair on a specific segment. 7. Each such pair occupies one segment, though not all segments in modern insects bear any visible appendages. Of all the insect orders, Orthoptera displays the greatest variety of features found in the heads of insects, including the sutures and sclerites. 6. Here, the vertex, or the apex dorsal region, is situated between the compound eyes of insects with hypognathous and opistognathous heads. In prognathous insects, the vertex is not found between the compound eyes, but rather where the ocelli are normally found. This is because the primary axis of the head is rotated 90 degrees to become parallel to the primary axis of the body. In some species, this region is modified and assumes a different name. 8.13. The ecdysial suture is made of the coronal, frontal, and epicranial sutures plus the ecdysial and cleavage lines, which vary among different species of insects. The ecdysial suture is longitudinally placed on the vertex, separating the epicranial halves of the head to the left and right sides. Depending on the insect, the suture may come in different shapes, like either A, Y, U or V. Those diverging lines that make up the ecdysial suture are called the frontal or frontogenal sutures. Not all species of insects have frontal sutures, but in those that do, the sutures split open during ecdysis, which provides an opening for the new instar to emerge from the integument. The frons is that part of the head capsule that lies ventrad or anterior of the vertex. The frons varies in size relative to the insect, and in many species, the definition of its borders is arbitrary, even in some insect taxa that have well-defined head capsules. In most species, though, the frons is bordered at its anterior by the frontoclypeal or epistomal sulcus above the clypeus. Laterally it is limited by the frontogenal sulcus, if present, and the boundary with the vertex, by the ecdysial cleavage line, if it is visible. If there is a median ocellus, it generally is on the frons, though in some insects such as many Hymenoptera, all 3 ocelli appear on the vertex. A more formal definition is that it is the sclerite from which the pharyngeal dilator muscles arise, but in many contexts that too is not helpful. 7. In the anatomy of some taxa, such as many Cicadomorpha, the front of the head is fairly clearly distinguished and tends to be broad and subvertical. That median area commonly is taken to be the frons. 9. The clypeus is a sclerite between the face and labrum, which is dorsally separated from the frons by the frontoclypeal suture in primitive insects. The The clipogenal suture laterally demarcates the clypeus, with the clypeus ventrally separated from the labrum by the clipolabral suture. The clypeus differs in shape and size, such as species of Lepidoptera with a large clypeus with elongated mouthparts. The cheek or genna forms the sclerotized area on each side of the head below the compound eyes extending to the gular suture. Like many parts making up the insect's head, the genna varies among species, with its boundaries difficult to establish. In dragonflies and damselflies, it is between the compound eyes, clypeus, and mouthparts. The postgenna is the area immediately posterior, or posterior or lower on the genna of pterygote insects, and forms the lateral and ventral parts of the occipital arch. The occipital arch is a narrow band forming the posterior edge of the head capsule arching dorsally over the foramen. The subgenal area is usually narrow, located above the mouthparts. This area also includes the hypostoma and fleurostoma. 813 to 14 The vertex extends anteriorly above the bases of the antennae as a prominent, pointed, concave rostrum. The posterior wall of the head capsule is penetrated by a large aperture, the foramen. Through it passes the organ systems, such as the nerve cord, esophagus, salivary ducts, and musculature, connecting the head with the thorax. 10. On the posterior aspect of the head are the occiput, postgenna, occipital foramen, posterior tentorial pit, bulla, postgenal bridge, hypostomal suture and bridge, and the mandibles, labium, and maxilla. The occipital suture is well founded in species of Orthoptera, but not so much in other orders. Where found, the occipital suture is the arched, horseshoe-shaped groove on the back of the head ending at the posterior of each mandible. The postoccipital suture is a landmark on the posterior surface of the head, and is typically near the occipital foramen. In pterygotes, the postocciput forms the extreme posterior, often U-shaped, which forms the rim of the head extending to the postoccipital suture. In pterygotes, such as those of Orthoptera, the occipital foramen and the mouth are not separated. The three types of occipital closures, or points under the occipital foramen that separate the two lower halves of the postgenna, are the hypostomal bridge, the postgenal bridge, and the gulla. The hypostomal bridge is usually found in insects with hypognathous orientation. The postgenal bridge is found in the adults of species of higher Diptera and Aculate Hymenoptera, while the gulla is found on some Coleoptera, Neuroptera, and Isoptera, which typically display prognathous-oriented mouthparts. 815. Compound eyes and ocelli. Most insects have one pair of large, prominent compound eyes composed of units called ommatidia, singular up to 30,000 in a single compound eye of, for example, large dragonflies. This type of eye gives less resolution than eyes found in vertebrates, but it gives an acute perception of movement and usually possesses UV and green sensitivity, and may have additional sensitivity peaks in other regions of the visual spectrum. Often an ability to detect the vector of polarized light exists in polarization of light. 11. There can also be an additional 2 or 3 ocelli, which help detect low light or small changes in light intensity. The image perceived is a combination of inputs from the numerous ommatidia, located on a convex surface, thus pointing in slightly different directions. Compared with simple eyes, compound eyes possess very large view angles and better acuity than the insect's dorsal ocelli, but some stemmatal equals larval eyes, for example, those of sophli larva tenthredinidae with an acuity of 4 degrees and very high polarization sensitivity, match the performance of compound eyes. 12-13. Ocellus cross-section. Compound eye cross-section. Because the individual lenses are so small, the effects of diffraction impose a limit on the possible resolution that can be obtained, assuming they do not function as phased arrays. This can only be countered by increasing lens size and number. To see with a resolution comparable to our simple eyes, humans would require compound eyes that would each reach the size of their heads. Compound eyes fall into 2 groups: apposition eyes, which form multiple inverted images, and superposition eyes, which form a single erect image. 14-15. Compound eyes grow at their margins with the addition of new ommatidia. 16. Antennae. Close-up of a fire ant, showing fine sensory hairs on antennae. Antennae, sometimes called feelers, are flexible appendages located on the insect's head which are used for sensing the environment. Insects can feel with their antennae because of the fine hairs, setae, that cover them. 17. 8-11. However, touch is not the only thing that antennae can detect. Numerous tiny sensory structures on the antennae allow insects to sense smells, temperature, humidity, pressure, and even potentially sense themselves in space. 17. 8-11. 18. 19. Some insects, including bees and some groups of flies, can also detect sound with their antennae. 20. The number of segments in an antenna varies amongst insects, with higher flies having 3 to 6 segments, 21, while adult cockroaches can have over 140. 22. The general shape of the antennae is also quite variable, but the first segment, the one attached to the head, is always called the scape, and the second segment is called the pedicel. The remaining antennal segments or flagellomers are called the flagellum. 17. 8-11. General insect antenna types are shown below. Types of insect antennae. 1. 17. 8-11. Aristate. Capitate. Clavate. Filiform. Flabellate. Geniculate. Cetaceous lamellate. Maniliform. Pectinate. Plumose. Serrate. Stellate. Mouthparts. The insect mouthparts consist of the maxilla, labium, and in some species, the mandibles. 816-23 The labrum is a simple, fused sclerite, often called the upper lip, and moves longitudinally. It is hinged to the clypeus. The mandible's jaws are a highly sclerotized pair of structures that move at right angles to the body, used for biting, chewing, and severing food. The maxillae are paired structures that can also move at right angles to the body and possess segmented palps. The labium, lower lip, is the fused structure that moves longitudinally and has a pair of segmented palps. 24. The development of insect mouthparts from the primitive chewing mouthparts of a grasshopper in the center A, to the lapping type B, of a bee, the siphoning type C, of a butterfly and the sucking type D, of a female mosquito. Legend. A. Antennae. C. Compound I. LB. Labium. LR. Labrum. MD. Mandibles. MX. Maxillae. The mouthparts and rest of the head can be articulated in at least 3 different positions: prognathous, opistognathous, and hypognathous. In species with prognathous articulation, the head is vertically aligned with the body, such as species of Formicidae, while in a hypognathous type, the head is aligned horizontally adjacent to the body. An opistognathous head is positioned diagonally, such as in species of Blatodia and some Coleoptera. 25 The mouthparts vary greatly between insects of different orders, but the 2 main functional groups are mandibulate and haustellate. Haustellate mouthparts are used for sucking liquids and can be further classified by the presence of stylets, which include piercing sucking, sponging, and siphoning. The stylets are needle-like projections used to penetrate plant and animal tissues. The stylets and the feeding tube form the modified mandibles, maxilla, and hypopharynx. 24. Mandibulate mouthparts, among the most common in insects, are used for biting and grinding solid foods. Piercing sucking mouthparts have stylets and are used to penetrate solid tissue and then suck up liquid food. Sponging mouthparts are used to sponge and suck liquids, and lack stylets, e.g. most Diptera. Siphoning mouthparts lack stylets and are used to suck liquids and are commonly found among species of Lepidoptera. Mandibular mouthparts are found in species of Odonata, adult Neuroptera, Coleoptera, Hymenoptera, Blatodia, Orthoptera, and Lepidoptera. However, most adult Lepidoptera have siphoning mouthparts, while their larvae, commonly called caterpillars, have mandibles. Mandibulate. The labrum is a broad lobe forming the roof of the preoral cavity, suspended from the clypeus in front of the mouth and forming the upper lip. 122-24 On its inner side, it is membranous and may be produced into a median lobe, the epipharynx, bearing some cincilla. The labrum is raised away from the mandibles by 2 muscles arising in the head and inserted medially into the anterior margin of the labrum. It is closed against the mandibles in part by 2 muscles arising in the head and inserted on the posterior lateral margins on 2 small sclerites, the tormae, and, at least in some insects, by a resolute spring in the cuticle at the junction of the labrum with the clypeus. 26 Until recently, the labrum generally was considered to be associated with the first head segment. However, recent studies of the embryology, gene expression, and nerve supply to the labrum show it is innervated by the tritocerebrum of the brain, which is the fused ganglia of the third head segment. This is formed from the fusion of parts of a pair of ancestral appendages found on the third head segment, showing their relationship. 122-24 Its ventral, or inner, surface is usually membranous and forms the lobe-like epipharynx, which bears mechanosencilla and chemisencilla. 27-28 Chewing insects have 2 mandibles, one on each side of the head. The mandibles are positioned between the labrum and maxillae. The mandibles cut and crush food, and may be used for defense. Generally, they have an apical cutting edge, and the more basal molar area grinds the food. They can be extremely hard, around 3 on Moss, or an indentation hardness of about 30 kilograms per square millimeter. Thus, many termites and beetles have no physical difficulty in boring through foils made from such common metals as copper, lead, tin, and zinc. 122-24 The cutting edges are typically strengthened by the addition of zinc, manganese, or rarely, iron, in amounts up to about 4% of the dry weight. 27 They are typically the largest mouthparts of chewing insects, being used to masticate, cut, tear, crush, chew food items. They open outwards to the sides of the head and come together medially. In carnivorous, chewing insects, the mandibles can be modified to be more knife-like, whereas in herbivorous chewing insects, they are more typically broad and flat on their opposing faces, e.g., caterpillars. In male stag beetles, the mandibles are modified to such an extent as to not serve any feeding function but are instead used to defend mating sites from other males. In ants, the mandibles also serve a defensive function, particularly in soldier castes. In bull ants, the mandibles are elongated and toothed, used as hunting and defensive appendages. Situated beneath the mandibles, paired maxillae manipulate food during mastication. Maxillae can have hairs and teeth along their inner margins. At the outer margin, the galea is a cupped or scoop-like structure, which sits over the outer edge of the labium. They also have palps, which are used to sense the characteristics of potential foods. The maxillae occupy a lateral position, one on each side of the head behind the mandibles. The proximal part of the maxilla consists of a basal cardo, which has a single articulation with the head, and a flat plate, the stipes, hinged to the cardo. Both cardo and stipes are loosely joined to the head by a membrane, so they are capable of movement. Distally on the stipes are 2 lobes, an inner lacinia, and an outer galea, one or both of which may be absent. More laterally on the stipes is a jointed, leg-like palp made up of many segments. In Orthoptera, there are 5. Anterior and posterior rotator muscles are inserted on the cardo, and ventral adductor muscles arising on the tentorium are inserted on both the cardo and stipes. Arising in the stipes are flexor muscles of the lacinium and galea, and another lacinial flexor arises in the cranium, but neither the lacinium nor the galea has an extensor muscle. The palp has levator and depressor muscles arising in the stipes, and each segment of the palp has a single muscle causing flexion of the next segment. 26. In mandibulate mouthparts, the labium is a quadrupedal structure, although it is formed from two fused secondary maxillae. It can be described as the floor of the mouth. With the maxillae, it assists with the manipulation of food during mastication or chewing, or, in the unusual case of the dragonfly nymph, extends out to snatch prey back to the head, where the mandibles can eat it. The labium is similar in structure to the maxilla, but with the appendages of the two sides fused by the midline, so they come to form a median plate. The basal part of the labium, equivalent to the maxillary cardines and possibly including a part of the sternum of the labial segment, is called the postmentum. This may be subdivided into a proximal submentum and a distal mentum. Distal to the postmentum, and equivalent to the fused maxillary stipets, is the prementum. The prementum closes the preoral cavity from behind. Terminally, it bears 4 lobes, 2 inner glossae, and 2 outer paraglossae, which are collectively known as the ligula. One or both pairs of lobes may be absent or they may be fused to form a single median process. A palp arises from each side of the prementum, often being 3-segmented. 26. The hypopharynx is a median lobe immediately behind the mouth, projecting forwards from the back of the preoral cavity. It is a lobe of uncertain origin, but perhaps associated with the mandibular segment. 26. In apterygotes, earwigs, and nymphal mayflies, the hypopharynx bears a pair of lateral lobes, the supralinguae singular, supralingua. It divides the cavity into a dorsal food pouch, or sabarium, and a ventral salivarium into which the salivary duct opens. 122-24. It is commonly found fused to the libium. 27. Most of the hypopharynx is membranous, but the odoral face is sclerotized distally, and proximally contains a pair of suspensory sclerites extending upwards to end in the lateral wall of the stomodium. Muscles arising on the frons are inserted into these sclerites, which distally are hinged to a pair of lingual sclerites. These, in turn, have inserted into them antagonistic pairs of muscles arising on the tentorium and labium. The various muscles serve to swing the hypopharynx forwards and back, and in the cockroach, 2 more muscles run across the hypopharynx and dilate the salivary orifice and expand the salivarium. 26. Diversity of mandibles. Piercing sucking. Mouth parts can have multiple functions. Some insects combine piercing parts along with sponging ones, which are then used to pierce through tissues of plants and animals. Female mosquitoes feed on blood, hemiphagus, making them disease vectors. The mosquito mouthparts consist of the proboscis, paired mandibles and maxillae. The maxillae form needle-like structures, called stylets, which are enclosed by the labium. When mosquito bites, maxillae penetrate the skin and anchor the mouthparts, thus allowing other parts to be inserted. The sheath-like labium slides back, and the remaining mouthparts pass through its tip and into the tissue. Then, through the hypopharynx, the mosquito injects saliva, which contains anticoagulants to stop the blood from clotting. And finally, the labrum, upper lip, is used to suck up the blood. Species of the genus Anopheles are characterized by their long palpi, 2 parts with widening end, almost reaching the end of labrum. 29. Diversity of piercing mouthparts. Mosquito. Cymex lectularius. Flea. Horsefly, female. Tick, Ixodes ricinus, an arachnid for comparison. Siphoning. The proboscis is formed from maxillary galleae and is an adaptation found in some insects for sucking. 30. The muscles of the sabarium or pharynx are strongly developed and form the pump. In Hemiptera and many Diptera, which feed on fluids within plants or animals, some components of the mouthparts are modified for piercing, and the elongated structures are called stylets. The combined tubular structures are referred to as the proboscis, although specialized terminology is used in some groups. In species of Lepidoptera, it consists of 2 tubes held together by hooks and separable for cleaning. Each tube is inwardly concave, thus forming a central tube through which moisture is sucked. Suction is effected by the contraction and expansion of a sac in the head. 31. The proboscis is coiled under the head when the insect is at rest and is extended only when feeding. 30. The maxillary palpi are reduced or even vestigial. 32. They are conspicuous and 5-segmented in some of the more basal families and are often folded. 8. The shape and dimensions of the proboscis have evolved to give different species wider and therefore more advantageous diets. 30. There is an allometric scaling relationship between the body mass of Lepidoptera and length of the proboscis. 33. From which an interesting adaptive departure is the unusually long-tongued hawk moth Xanthopon morgani praedicta. Charles Darwin predicted the existence and proboscis length of this moth before its discovery based on his knowledge of the long-spurred Madagascan star orchid Angraecum sesquipedale. 34. Siphoning mouthparts. Sponging. The mouthparts of insects that feed on fluids are modified in various ways to form a tube through which liquid can be drawn into the mouth and usually another through which saliva passes. The muscles of the sabarium or pharynx are strongly developed to form a pump. 20. In Brachycera, the labellum is especially prominent and used for sponging liquid or semi-liquid food. 35. The labella are a complex structure consisting of many grooves, called pseudotracheae, which sop up liquids. Salivary secretions from the labella assist in dissolving and collecting food particles so they can be more easily taken up by the pseudotracheae or lay their egg on the suitable media. This is thought to occur by capillary action. The liquid food is then drawn up from the pseudotracheae through the food channel into the esophagus. 36. The mouthparts of bees are of a chewing and lapping sucking type. Lapping is a mode of feeding in which liquid or semi-liquid food adhering to a protrusible organ, or tongue, is transferred from substrate to mouth. In the honeybee Hymenoptera, Apidae, Apis mellifera, the elongated and fused labial glossae form a hairy tongue, which is surrounded by the maxillary galleae and the labial palps to form a tubular proboscis containing a food canal. In feeding, the tongue is dipped into the nectar or honey, which adheres to the hairs, and then is retracted so the adhering liquid is carried into the space between the galleae and labial palps. This back and forth glossal movement occurs repeatedly. Movement of liquid to the mouth results from the action of the sebareal pump, facilitated by each retraction of the tongue pushing liquid up the food canal either for feeding requirements or to have a suitable media for laying their egg. Movement of liquid to the mouth results from the action of the sebareal 122-24. Sponging mouthparts. Thorax. The insect thorax has three segments: the prothorax, mesothorax, and metathorax. The anterior segment, closest to the head, is the prothorax. Its major features are the first pair of legs and the pronotum. The middle segment is the mesothorax. Its major features are the second pair of legs and the anterior wings, if any. The third, the posterior, thoracic segment, abutting the abdomen, is the metathorax, which bears the third pair of legs and the posterior wings. Each segment is delineated by an intersegmental suture. Each segment has four basic regions. The dorsal surface is called the tergum, or notum, to distinguish it from the abdominal terga. 122-24. The two lateral regions are called the pleura, singular, pleuron, and the ventral aspect is called the sternum. In turn, the notum of the prothorax is called the pronotum, the notum for the mesothorax is called the mesonotum, and the notum for the metathorax is called the metanotum. Continuing with this logic, there are also the mesopelora and metapelora, as well as the mesosternum and metasternum. 8. The tergal plates of the thorax are simple structures in apterygotes and many immature insects but are variously modified in winged adults. The pterothoracic nota each have two main divisions: the anterior, wing-bearing allenotum, and the posterior, phragma-bearing postnotum. Phragmata, singular. Phragma are plate-like apodoms that extend inwards below the antecostal sutures, marking the primary intersegmental folds between segments. Phragmata provide attachment for the longitudinal flight muscles. Each allenotum, sometimes confusingly referred to as a notum, may be traversed by sutures that mark the position of internal strengthening ridges and commonly divide the plate into three areas: the anterior prescutum, the scutum, and the smaller posterior scutellum. The lateral pleural sclerites are believed to be derived from the subcoxal segment of the ancestral insect leg. These sclerites may be separate, as in silverfish, or fused into an almost continuous sclerotic area, as in most winged insects. 122-24. Prothorax. The pronotum of the prothorax may be simple in structure and small in comparison with the other nota, but in beetles, mantids, many bugs, and some orthoptera, the pronotum is expanded, and in cockroaches, it forms a shield that covers part of the head and mesothorax. 8:122-24. Pterothorax. Because the mesothorax and metathorax hold the wings, they have a combined name called the pterothorax. Pteron equals wing. The forewing, which goes by different names in different orders, e.g., the tegmina in Orthoptera and elytra in Coleoptera, arises between the mesonotum and the mesopeloron, and the hindwing articulates between the metanotum and metapeloron. The legs arise from the mesopeloron and metapelora. The mesothorax and metathorax each have a pleural suture, mesopleural and metapleural sutures, that runs from the wing base to the coxa of the leg. The sclerite anterior to the pleural suture is called the episternum, serially, the mesepisternum and metapisternum. The sclerite posterior to the suture is called the epimyron, serially, the mesepimyron and metapimyron. Spiracles, the external organs of the respiratory system, are found on the pterothorax, usually one between the pro and mesopleoron, as well as one between the meso and metapeloron. 8. The ventral view or sternum follows the same convention, with the prosternum under the prothorax, the mesosternum under the mesothorax, and the metasternum under the metathorax. The notum, pleura, and sternum of each segment have a variety of different sclerites and sutures, varying greatly from order to order, and they will not be discussed in detail in this section. 8. Wings. Most phylogenetically advanced insects have two pairs of wings located on the second and third thoracic segments. 122-24. Insects are the only invertebrates to have developed flight capability, and this has played an important part in their success. Insect flight is not very well understood, relying on turbulent aerodynamic effects. The primitive insect groups use muscles that act directly on the wing structure. The more advanced groups making up the Neoptera have foldable wings, and their muscles act on the thorax wall and power the wings indirectly. 122-24. These muscles can contract multiple times for each single nerve impulse, allowing the wings to beat faster than would ordinarily be possible. Insect flight can be rapid, maneuverable, and versatile, possibly due to the changing shape, extraordinary control, and variable motion of the insect wing. Insect orders use different flight mechanisms. For example, the flight of a butterfly can be explained using steady state, non-transitory aerodynamics, and thin airfoil theory. Cross-section of wing vein. Internal. Each of the wings consists of a thin membrane supported by a system of veins. The membrane is formed by two layers of integument closely opposed, while the veins are formed where the two layers remain separate and the cuticle may be thicker and more heavily sclerotized. Within each of the major veins is a nerve and a trachea, and, since the cavities of the veins are connected with the hemocoel, hemolymph can flow into the wings. 26. As the wing develops, the dorsal and ventral integumental layers become closely opposed over most of their area, forming the wing membrane. The remaining areas form channels, the future veins, in which the nerves and tracheae may occur. The cuticle surrounding the veins becomes thickened and more heavily sclerotized to provide strength and rigidity to the wing. Hairs of two types may occur on the wings: microtrichia, which are small and irregularly scattered, and macrotrichia, which are larger, socketed, and may be restricted to veins. The scales of Lepidoptera and Trichoptera are highly modified macrotrichia. 27. Veins. Venation of insect wings, based on the Comstock-Needham system. In some minuscule insects, the venation may be reduced. In chalcidoid wasps, for instance, only the subcosta and part of the radius are present. Conversely, an increase in venation may occur by the branching of existing veins to produce accessory veins or by the development of additional, intercalary veins between the original ones, as in the wings of Orthoptera, grasshoppers and crickets. Large numbers of cross veins are present in some insects, and they may form a reticulum as in the wings of Odonata, dragonflies and damselflies, and at the base of the forewings of Tetigonioidea and Acridoidea, katydids and grasshoppers, respectively. 26. The Archetoctian is the name given to a hypothetical scheme of wing venation proposed for the very first winged insect. It is based on a combination of speculation and fossil data. Since all winged insects are believed to have evolved from a common ancestor, the Archetoctian represents the template that has been modified and streamlined by natural selection for 200 million years. According to current dogma, the Archetoctian contains 6 to 8 longitudinal veins. These veins and their branches are named according to a system devised by John Comstock and George Needham, the Comstock-Needham system. 37. Costa C, the leading edge of the wing. Subcosta SC, second longitudinal vein, behind the costa, typically unbranched, radius R, third longitudinal vein. 1 to 5 branches reach the wing margin, media M, fourth longitudinal vein. 1 to 4 branches reach the wing margin, cubitus Q, fifth longitudinal vein. 1 to 3 branches reach the wing margin, anal veins, A1, A2, A3, unbranched veins behind the cubitus. The costa C is the leading marginal vein on most insects, although a small vein, the precosta, is sometimes found above the costa. In almost all extant insects, 141 to 42, the precosta is fused with the costa. The costa rarely ever branches because it is at the leading edge, which is associated at its base with the humeral plate. The trachea of the costal vein is perhaps a branch of the subcostal trachea. Located after the costa is the third vein, the subcosta, which branches into 2 separate veins, the anterior and posterior. The base of the subcosta is associated with the distal end of the neck of the first axillary. The fourth vein is the radius, which is branched into 5 separate veins. The radius is generally the strongest vein of the wing. Toward the middle of the wing, it forks into a first undivided branch, R1, and a second branch, called the radial sector, RA, which subdivides dichotomously into 4 distal branches, R2, R3, R4, R5. Basally, the radius is flexibly united with the anterior end of the second axillary, 2-ax. 38. The fifth vein of the wing is the media. In the archetype pattern A, the media forks into 2 main branches, a media anterior, MA, which divides into 2 distal branches, MA1, MA2, and a median sector, or media posterior, MP, which has 4 terminal branches, M1, M2, M3, M4. In most modern insects, the media anterior has been lost, and the usual media is the 4-branched media posterior with the common basal stem. In the Ephemerida, according to present interpretations of the wing venation, both branches of the media are retained, while in Odonata, the persisting media is the primitive anterior branch. The stem of the media is often united with the radius, but when it occurs as a distinct vein, its base is associated with the distal median plate, M, or is continuously sclerotized with the latter. The cubitus, the 6th vein of the wing, is primarily 2-branched. The primary forking takes place near the base of the wing, forming the 2 principal branches, Q1, Q2. The anterior branch may break up into several secondary branches, but commonly it forks into 2 distal branches. The second branch of the cubitus, Q2, in Hymenoptera, Trichoptera, and Lepidoptera, was mistaken by Comstock and Needham for the first anal. Proximally, the main stem of the cubitus is associated with the distal median plate, M, of the wing base. 38. The postcubitus, PCU, is the first anal of the Comstock and Needham system. The postcubitus, however, has the status of an independent wing vein and should be recognized as such. In nymphal wings, its trachea arises between the cubital trachea and the group of vanal tracheae. In the mature wings of more generalized insects, the postcubitus is always associated proximally with the cubitus and is never intimately connected with the flexor sclerite, 3-ax, of the wing base. In Neuroptera, Mecoptera, and Trichoptera, the postcubitus may be more closely associated with the vanal veins, but its base is always free from the latter. The postcubitus is usually unbranched. Primitively, it is 2-branched. The vanal veins, LV to NB, are the anal veins immediately associated with the third axillary, and are directly affected by the movement of this sclerite that brings about the flexion of the wings. In number, the vanal veins vary from 1 to 12, according to the expansion of the vanal area of the wing. The vanal tracheae usually arise from a common tracheal stem in nymphal insects, and the veins are regarded as branches of a single anal vein. Distally, the vanal veins are either simple or branched. The jugal vein, J, of the jugal lobe of the wing is often occupied by a network of irregular veins, or it may be entirely membranous. Sometimes it contains 1 or 2 distinct, small veins, the first jugal vein, or vena arcuata, and the second jugal vein, or vena cardinalis, 2J. 38. CSC cross veins, run between the costa and subcosta. R cross veins, run between adjacent branches of the radius. RM cross veins, run between the radius and media. MQ cross veins, run between the media and cubitus. All the veins of the wing are subject to secondary forking and union by cross veins. In some orders of insects, the cross veins are so numerous, the whole venational pattern becomes a close network of branching veins and cross veins. Ordinarily, however, a definite number of cross veins having specific locations occurs. The more constant cross veins are the humeral cross vein H between the costa and subcosta, the radial cross vein R between R and the first fork of Rs, the sectorial cross vein S between the two forks of R8, the median cross vein MM between M2 and M3, and the mediocubital cross vein MQ between the media and the cubitus. 38. The veins of insect wings are characterized by a convex-concave placement, such as those seen in mayflies, i.e., concavas, down, and convexas, up, which alternate regularly and by their branching. Whenever a vein forks, there is always an interpolated vein of the opposite position between the two branches. The concave vein will fork into two concave veins, with the interpolated vein being convex, and the regular alteration of the veins is preserved. 39. The veins of the wing appear to fall into an undulating pattern according to whether they tend to fold up or down when the wing is relaxed. The basal shafts of the veins are convex, but each vein forks distally into an anterior convex branch and a posterior concave branch. Thus, the costa and subcosta are regarded as convex and concave branches of a primary first vein. Rs is the concave branch of the radius, posterior media is the concave branch of the media. Q1 and Q2 are respectively convex and concave, while the primitive postcubitus and the first vanal have each an anterior convex branch and a posterior concave branch. The convex or concave nature of the veins has been used as evidence in determining the identities of the persisting distal branches of the veins of modern insects, but it has not been demonstrated to be consistent for all wings. 2638. Fields. Wing areas are delimited and subdivided by fold lines, along which the wings can fold, and flexion lines, which flex during flight. Between the flexion and the fold lines, the fundamental distinction is often blurred, as fold lines may permit some flexibility or vice versa. Two constants, found in nearly all insect wings, are the cloval, a flexion line, and jugal folds, or fold line, forming variable and unsatisfactory boundaries. Wing folding can be very complicated, with transverse folding occurring. Insect morphology. Insect morphology. Antenna biology. Antennae, SG. Antennae, sometimes referred to as feelers, are paired appendages used for sensing in arthropods. Large antennae on a longhorn beetle. Antennae are connected to the first one or two segments of the arthropod head. They vary widely in form but are always made of one or more jointed segments. While they are typically sensory organs, the exact nature of what they sense and how they sense it is not the same in all groups. Functions may variously include sensing touch, air motion, heat, vibration, sound, and especially smell or taste. 1-2 antennae are sometimes modified for other purposes, such as mating, brooding, swimming, and even anchoring the arthropod to a substrate. 2 larval arthropods have antennae that differ from those of the adult. Many crustaceans, for example, have free-swimming larvae that use their antennae for swimming. Antennae can also locate other group members if the insect lives in a group, like the ant. The common ancestor of all arthropods likely had one pair of uniramous, unbranched, antenna-like structures, followed by one or more pairs of biramus, having two major branches, leg-like structures, as seen in some modern crustaceans and fossil trilobites. 3. Except for the chelicerates and proterans, which have none, all non-crustacean arthropods have a single pair of antennae. 4. Crustaceans. Cutaway diagram of a barnacle, with antennae highlighted by arrow. Crustaceans bear two pairs of antennae. The pair attached to the first segment of the head are called primary antennae or antennules. This pair is generally uniramous, but is biramus in crabs and lobsters and remipedes. The pair attached to the second segment are called secondary antennae or simply antennae. The second antennae are plesiomorphically biramus, but many species later evolved uniramous pairs. 2. The second antennae may be significantly reduced, e.g. remipedes, or apparently absent, e.g. barnacles. The subdivisions of crustacean antennae have many names, including flagellomers, a shared term with insects, annuli, articles, and segments. The terminal ends of crustacean antennae have two major categorizations, segmented and flagellate. An antenna is considered segmented if each of the annuli is separate from those around it and has individual muscle attachments. Flagellate antennae, on the other hand, have muscle attachments only around the base, acting as a hinge for the flagellum, a flexible string of annuli with no muscle attachment. 5. There are several notable non-sensory uses of antennae in crustaceans. Many crustaceans have a mobile larval stage called anopleus, which is characterized by its use of antennae for swimming. Barnacles, a highly modified crustacean, use their antennae to attach to rocks and other surfaces. 2. The second antennae in the burrowing Hippoidea and Choristidae have setae that interlock to form a tube or snorkel, which funnels filtered water over the gills. 6. A spiny lobster, showing the enlarged second antennae. The large flattened plates in front of the eyes of a slipper lobster are the modified second antennae. The crab Cancer pygurus, showing its reduced antennae. Antennules of the Caribbean hermit crab. The nauplius larva of a shrimp with antennae. Used for swimming. Examples of crustacean antennae. Copepoda, isopoda, amphipoda, decapita, remipedea, cirripedia. P. rem. Aryantannae. Secondar ntennae. Insects. Terms used to describe shapes of insect antennae. Antennal shape in the Lepidoptera from C. T. Bingham, 1905. Some claim insects evolved from prehistoric crustaceans, and they have secondary antennae-like crustaceans, but not primary antennae. Antennae are the primary olfactory sensors of insects, 7, and are accordingly well equipped with a wide variety of sensilla, singular, sensillum. Paired, mobile, and segmented, they are located between the eyes on the forehead. Embryologically, they represent the appendages of the second head segment. 8. All insects have antennae, however they may be greatly reduced in the larval forms. Amongst the non-insect classes of the hexapoda, both Collembola and Dipleura have antennae, but Protura do not. 9. Antennal fibrillae play an important role in Culex pipiens mating practices. The erection of these fibrillae is considered to be the first stage in reproduction. These fibrillae serve different functions across the sexes. As antennal fibrillae are used by female C. pipiens to locate hosts to feed on, male C. pipiens utilize them to locate female mates. Structure. Electron micrograph of antenna surface detail of a wasp, Vespula vulgaris. The three basic segments of the typical insect antenna are the scape or scapus base, the pedicel or pedicellus stem, and finally the flagellum, which often comprises many units known as flagellomers. 10. The pedicel, the second segment, contains the Johnston's organ, which is a collection of sensory cells. 11. The scape is mounted in a socket in a more or less ring-shaped sclerotized region called the torulus, often a raised portion of the insect's head capsule. The socket is closed off by the membrane into which the base of the scape is set. However, the antenna does not hang free on the membrane, but pivots on a rigidly sprung projection from the rim of the torulus. That projection on which the antenna pivots is called the antennifer. The whole structure enables the insect to move the antenna as a whole by applying internal muscles connected to the scape. The pedicel is flexibly connected to the distal end of the scape, and its movements in turn can be controlled by muscular connections between the scape and pedicel. The number of flagellomers can vary greatly between insect species, and often is of diagnostic importance. True flagellomers are connected by membranous linkage that permits movement, though the flagellum of true insects does not have any intrinsic muscles. Some other arthropoda do, however, have intrinsic muscles throughout the flagellum. Such groups include the Symphyla, Collembola, and Dipleura. In many true insects, especially the more primitive groups such as Thysanura and Blatodia, the flagellum partly or entirely consists of a flexibly connected string of small ring-shaped annuli. The annuli are not true flagellomers, and in a given insect species the number of annuli generally is not as consistent as the number of flagellomers in most species. 10. In many beetles and in the chalcidoid wasps, the apical flagellomers form a club shape, called the clava. The collective term for the segments between the club and the antennal base is the funicle. Traditionally, in describing beetle anatomy, the term funicle refers to the segments between the club and the scape. However, traditionally in working on wasps, the funicle is taken to comprise the segments between the club and the pedicel. 10. Quite commonly, the funicle beyond the pedicel is quite complex in Endopterygoda such as beetles, moths, and Hymenoptera, and one common adaptation is the ability to fold the antenna in the middle, at the joint between the pedicel and the flagellum. This gives an effect like a knee bend, and such an antenna is said to be geniculate. Geniculate antennae are common in the Coleoptera and Hymenoptera. They are important for insects like ants that follow scent trails, for bees and wasps that need to sniff the flowers that they visit, and for beetles such as Scarabaeidae and Curculionidae that need to fold their antennae away when they self-protectively fold up all their limbs in defensive attitudes. Because the funicle is without intrinsic muscles, it generally must move as a unit, in spite of being articulated. However, some funicles are complex and very mobile. For example, the Scarabaeidae have lamellate antennae that can be folded tightly for safety or spread openly for detecting odors or pheromones. The insect manages such actions by changes in blood pressure, by which it exploits elasticity in walls and membranes in the funicles, which are in effect erectile. 12. In the groups with more uniform antennae, for example, millipedes, all segments are called antennomers. Some groups have a simple or variously modified apical or subapical bristle called an arista. This may be especially well developed in various Diptera. 13. Functions. Olfactory receptors, scales and holes, on the antenna of the butterfly Aglae eo, electron micrograph. Olfactory receptors on the antennae bind to free-floating molecules, such as water vapor, and odors including pheromones. The neurons that possess these receptors signal this binding by sending action potentials down their axons to the antennal lobe in the brain. From there, neurons in the antennal lobes connect to mushroom bodies that identify the odor. The sum of the electrical potentials of the antennae to a given odor can be measured using an electroantennogram. 14. In the monarch butterfly, antennae are necessary for proper time-compensated solar compass orientation during migration. Antennal clocks exist in monarchs, and they are likely to provide the primary timing mechanism for sun compass orientation. 15. In the African cotton leafworm, antennae have an important function in signaling courtship. Specifically, antennae are required for males to answer the female mating call. Although females do not require antennae for mating, a mating that resulted from a female without antennae was abnormal. 16. In the diamondback moth, antennae serve to gather information about a host plant's taste and odor. After the desired taste and odor has been identified, the female moth will deposit her eggs onto the plant. 17. Giant swallowtail butterflies also rely on antenna sensitivity to volatile compounds to identify host plants. It was found that females are actually more responsive with their antenna sensing, most likely because they are responsible for oviposition on the correct plant. 18. In the crepuscular hawk moth, Manduca sexta, antennae aid in flight stabilization. Similar to halteres in dipteran insects, the antennae transmit Coriolis forces through the Johnston's organ that can then be used for corrective behavior. A series of low-light, flight stability studies in which moths with flagella amputated near the pedicel showed significantly decreased flight stability over those with intact antennae. 19. To determine whether there may be other antennal sensory inputs, a second group of moths had their antennae amputated and then reattached, before being tested in the same stability study. These moths showed slightly decreased performance from intact moths, indicating there are possibly other sensory inputs used in flight stabilization. Re-amputation of the antennae caused a drastic decrease in flight stability to match that of the first amputated group. References. Structure. Electron micrograph of antenna surface detail of a wasp, Vespula vulgaris. The three basic segments of the typical insect antenna are the scape or scapus base, the pedicel or pedicellus stem, and finally the flagellum, which often comprises many units known as flagellomers. 10. The pedicel, the second segment, contains the Johnston's organ, which is a collection of sensory cells. 11. The scape is mounted in a socket in a more or less ring-shaped sclerotized region called the torulus, often a raised portion of the insect's head capsule. The socket is closed off by the membrane into which the base of the scape is set. However, the antenna does not hang free on the membrane, but pivots on a rigidly sprung projection from the rim of the torulus. That projection on which the antenna pivots is called the antennifer. The whole structure enables the insect to move the antenna as a whole by applying internal muscles connected to the scape. The pedicel is flexibly connected to the distal end of the scape, and its movements in turn can be controlled by muscular connections between the scape and pedicel. The number of flagellomers can vary greatly between insect species, and often is of diagnostic importance. True flagellomers are connected by membranous linkage that permits movement, though the flagellum of true insects does not have any intrinsic muscles. Some other arthropoda do, however, have intrinsic muscles throughout the flagellum. Such groups include the Symphyla, Collembola, and Dipleura. In many true insects, especially the more primitive groups such as Thysanura and Blatodia, the flagellum partly or entirely consists of a flexibly connected string of small ring-shaped annuli. The annuli are not true flagellomers, and in a given insect species the number of annuli generally is not as consistent as the number of flagellomers in most species. 10. In many beetles and in the chalcidoid wasps, the apical flagellomers form a club shape, called the clava. The collective term for the segments between the club and the antennal base is the funicle. Traditionally, in describing beetle anatomy, the term funicle refers to the segments between the club and the scape. However, traditionally in working on wasps, the funicle is taken to comprise the segments between the club and the pedicel. 10. Quite commonly, the funicle beyond the pedicel is quite complex in Endopterygota such as beetles, moths, and Hymenoptera, and one common adaptation is the ability to fold the antenna in the middle, at the joint between the pedicel and the flagellum. This gives an effect like a knee bend, and such an antenna is said to be geniculate. Geniculate antennae are common in the Coleoptera and Hymenoptera. They are important for insects like ants that follow scent trails, for bees and wasps that need to sniff the flowers that they visit, and for beetles such as Scarabaeidae and Curculionidae that need to fold their antennae away when they self-protectively fold up all their limbs in defensive attitudes. Because the funicle is without intrinsic muscles, it generally must move as a unit, in spite of being articulated. However, some funicles are complex and very mobile. For example, the Scarabaeidae have limellate antennae that can be folded tightly for safety or spread openly for detecting odors or pheromones. The insect manages such actions by changes in blood pressure, by which it exploits elasticity in walls and membranes in the funicles, which are in effect erectile. 12. In the groups with more uniform antennae, for example, millipedes, all segments are called antennomers. Some groups have a simple or variously modified apical or subapical bristle called an arista. This may be especially well developed in various Diptera. 13. Functions. Olfactory receptors, scales and holes, on the antenna of the butterfly Aglaeo, electron micrograph. Olfactory receptors on the antennae bind to free-floating molecules, such as water vapor, and odors including pheromones. The neurons that possess these receptors signal this binding by sending action potentials down their axons to the antennal lobe in the brain. From there, neurons in the antennal lobes connect to mushroom bodies that identify the odor. The sum of the electrical potentials of the antennae to a given odor can be measured using an electroantennogram. 14. In the monarch butterfly, antennae are necessary for proper time-compensated solar compass orientation during migration. Antennal clocks exist in monarchs, and they are likely to provide the primary timing mechanism for sun compass orientation. The neurons. Olfactory receptors on the antennae bind to free-floating molecules, such as water vapor, and odors including pheromones. The neurons that possess these receptors signal this binding by sending action potentials down their axons to the antennal lobe in the brain. From there, neurons in the antennal lobes connect to mushroom bodies that identify the odor. The sum of the electrical potentials of the antennae to a given odor can be measured using an electroantennogram. 14. In the monarch butterfly, antennae are necessary for proper time-compensated solar compass orientation during migration. Antennal clocks exist in monarchs, and they are likely to provide the primary timing mechanism for sun compass orientation. 15. In the African cotton leafworm, antennae have an important function in signaling courtship. Specifically, antennae are required for males to answer the female mating call. Although females do not require antennae for mating, a mating that resulted from a female without antennae was abnormal. 16. In the diamondback moth, antennae serve to gather information about a host plant's taste and odor. After the desired taste and odor has been identified, the female moth will deposit her eggs onto the plant. 17. Giant swallowtail butterflies also rely on antenna sensitivity to volatile compounds to identify host plants. It was found that females are actually more responsive with their antenna sensing, most likely because they are responsible for oviposition on the correct plant. 18. In the crepuscular hawk moth, Manduca sexta, antennae aid in flight stabilization. Similar to halteres in dipteran insects, the antennae transmit Coriolis forces through the Johnston's organ that can then be used for corrective behavior. A series of low-light, flight stability studies in which moths with flagally amputated near the pedicel showed significantly decreased flight stability over those with intact antennae. 19. To determine whether there may be other antennal sensory inputs, a second group of moths had their antennae amputated and then reattached, before being tested in the same stability study. These moths showed slightly decreased performance from intact moths, indicating there are possibly other sensory inputs used in flight stabilization. Re-amputation of the antennae caused a drastic decrease in flight stability to match that of the first amputated group. References. In the diamondback moth, antennae serve to gather information. From there, neurons in the antennal lobes connect to mushroom bodies that identify the odor. The sum of the electrical potentials of the antennae to a given odor can be measured using an electroantennogram. 14. In the monarch butterfly, antennae are necessary for proper time-compensated solar compass orientation during migration. Antennal clocks exist in monarchs, and they are likely to provide the primary timing mechanism for sun compass orientation. 15. In the African cotton leafworm, antennae have an important function in signaling courtship. Specifically, antennae are required for males to answer the female mating call. Although females do not require antennae for mating, a mating that resulted from a female without antennae was abnormal. 16. In the diamondback moth, antennae serve to gather information about a host plant's taste and odor. After the desired taste and odor has been identified, the female moth will deposit her eggs onto the plant. 17. Giant swallowtail butterflies also rely on antenna sensitivity to volatile compounds to identify host plants. It was found that females are actually more responsive with their antenna sensing, most likely because they are responsible for oviposition on the correct plant. 18. In the crepuscular hawk moth, Manduca sexta, antennae aid in flight stabilization. From there, neurons in the antennal lobes connect to mushroom bodies that identify the odor. The sum of the electrical potentials of the antennae to a given odor can be measured using an electroantennogram. 14. In the monarch butterfly, antennae are necessary for proper time-compensated solar compass orientation during migration. Antennal clocks exist in monarchs, and they are likely to provide the primary timing mechanism for sun compass orientation. 15. In the African cotton leafworm, antennae have an important function in signaling courtship. Specifically, antennae are required for males to answer the female mating call. Although females do not require antennae for mating, a mating that resulted from a female without antennae was abnormal. 16. In the diamondback moth, antennae serve to gather information about a host plant's taste and odor. After the desired taste and odor has been identified, the female moth will deposit her eggs onto the plant. 17. Giant swallowtail butterflies also rely on antenna sensitivity to volatile compounds to identify host plants. It was found that females are actually more responsive with their antenna sensing, most likely because they are responsible for oviposition on the correct plant. 18. In the crepuscular hawk moth, Manduca sexta, antennae aid in flight stabilization. Similar to halteres in dipteran insects, the antennae transmit Coriolis forces through the Johnston's organ that can then be used for corrective behavior. A series of low-light, flight stability studies in which moths with flagally amputated near the pedicel showed significantly decreased flight stability over those with intact antennae. 19. To determine whether there may be other antennal sensory inputs, a second group of moths had their antennae amputated and then reattached, before being tested in the same stability study. These moths showed slightly decreased performance from intact moths. In the crepuscular, it was found that females are actually more responsive with their antenna sensing, most likely because they are responsible for oviposition on the correct plant. 18. In the crepuscular hawk moth, Manduca sexta, antennae aid in flight stabilization. Similar to halteres in dipteran insects, the antennae transmit Coriolis forces through the Johnston's organ that can then be used for corrective behavior. A series of low-light, flight stability studies in which moths with flagally amputated near the pedicel showed significantly decreased flight stability over those with intact antennae. 19. To determine whether there may be other antennal sensory inputs, a second group of moths had their antennae amputated and then reattached, before being tested in the same stability study. These similar 14. In the monarch butterfly, antennae are necessary for proper time-compensated solar compass orientation during migration. Antennal clocks exist in monarchs, and they are likely to provide the primary timing mechanism for sun compass orientation. 14. In the monarch butterfly, antennae are necessary for proper time-compensated solar compass orientation during migration. Antennal clocks exist in monarchs, and they are likely to provide the primary timing mechanism for sun compass orientation. 15. In the African cotton leafworm, antennae have an important function in signaling courtship. Specifically, antennae are required for males to answer the female mating call. Although females do not require antennae for mating, a mating that resulted from a female without antennae was abnormal. 16. In the diamondback moth, antennae serve to gather information about a host plant's taste and odor. After the desired taste and odor has been identified, the female moth will deposit her eggs onto the plant. 17. Giant swallowtail butterflies also rely on antenna sensitivity to volatile compounds to identify host plants. It was found that females are actually more responsive with their antenna sensing, most likely because they are responsible for oviposition on the correct plant. 18. In the crepuscular hawk moth, Manduca sexta, antennae aid in flight stabilization. Similar to halteres in dipteran insects, the antennae transmit Coriolis forces through the Johnston's organ that can then be used for corrective behavior. A series